So good afternoon, everyone, and thanks very much for visiting Szeged and for participating at this event. I hope you will enjoy this. So I also come from the Biological Research Center of the Hungarian Academy of Sciences. So this is not part of the university, but we work very closely with a lot of links with the university. As a matter of fact, we have, I think, on the order of 100 or 200 students, PhD students within this institute. So we already seen a spaghetti slide. This is something similar. So really these two pictures represent like the extreme end of the spectrum of human diet that one can have, like soybean and beef burger. And what I find particularly striking is that there is something deep in common in those two food types, is that basically our body can build up their own molecules from either of them. So basically, we might not get exactly the same shape of body, but clearly we are able to utilize these nutrients from a large number of nutrient sources. And what is behind this is the process called metabolism, which means that the cell takes up food molecules, breaks it down into energy and some very small building blocks that can be assembled into their own macromolecules, like their own DNA, lipids, proteins, and so on and so forth. So behind this kind of magical process is an intricate and complex network of biochemical reactions, which contains in human body 5,000–6,000 known biochemical reactions, which basically an elementary step in this network is a single chemical reaction converting one or more molecules into something else. So basically, these are the key players. The metabolites, what we call them, are the key players of cellular metabolism. Metabolism has been studied for many, many decades, and one kind of emerging picture is that the structure of the metabolic network looks pretty similar, even across very highly dissimilar species. Just to quote Jacques Monod here, so what is true for an elephant, what is true for E. coli is also true for the elephant, would really well apply to at least to central pathways of metabolism. But if you look at the details, how these metabolic networks work, then actually there are large differences. And nowadays, there are some really nice technologies to measure the behavior of these networks. These are called metabolomics approaches, which basically is about breaking up cells, getting all the small molecule components, metabolites out of them, and using either mass spectrometry or some other methods, measure the concentration of these metabolites, and we can compare it between conditions, tissues, organisms. So today we can measure like 100 to 300 metabolites in a single experimental run. And also these techniques are now pretty cheap, so it can be scaled up to measure metabolome profile, as what we call the concentration levels measured across all metabolites. It can now be done for human populations, large populations, for thousands of people. And actually, these studies reveal that there are quite some variation between how the metabolism of those sitting in this room, for example, behave. And some of them are already kind of linked to known diseases. Just think about cholesterol level and some other kind of harmful metabolites. So my lab, one of the major goals of my lab is to understand better what drives these differences between species and between individuals. What are the evolutionary forces? And clearly some of these differences might be beneficial. For example, it has been shown that some brain lipids are very different in chimp and human, and it has been linked to some cognitive capabilities. Some others are, like what I mentioned earlier, Some are linked to diseases. So if you have a higher cholesterol level, it predisposes you to certain diseases. But many of these changes might not matter at all, what we call in evolutionary biology as neutral, might not have functional consequences. But in general, we have examples of all of these, but in general, we just don't understand what metabolic differences would give rise to functional or non-functional differences in general. So, and the related question, of course, is that if we would understand better, if we could understand better what drives metabolic differences, then we might use it to increase the accuracy of diagnosis. I will come back to that later. So what are the tools that we use to study these questions? On the one hand, we are running actually, we set up a metabolomics lab in the institute and measure metabolites directly, actually mostly from unicellular organisms. On the other hand, and as far as I know, this is probably the first place in Hungary where kind of large-scale metabolomics is done, so more than a few dozen, up to several hundred metabolites can be quantified. And on the other hand, what actually is kind of the major activity of the lab is to do bioinformatics analysis, both of our own data and a lot of published data. There are a lot of published data for human, for example, and even for several mammals that can be used. And to use this data, analyze in the context of the metabolic network to understand how these metabolite level changes might translate into functional consequences. As a matter of fact, if I can give you one advice, if you are really into biological sciences, then learn programming, just at the very basic level. But for me, that was probably the most useful skill I got during high school, because there are so much data out there. In almost all fields in biology today, you generate an order of magnitude more data than just 5–10 years ago, and there is just no way you can analyze them manually or just by looking at them by eye. So one of the major aims we pursuing is to understand, to try to identify those metabolites and those parts of metabolisms that really matter during evolution, that selection really cares about. These are the most conserved ones, and probably they are functionally the most important ones. So as an analogy, one can use evolutionary approaches to find these functionally important places or functionally important changes. And as an analogy, in genomic sciences, when the DNA sequence is determined for different organisms, it's quite commonplace to then line up them and compare the sequences across different species and identify those sites, those letters in the DNA, which do not tend to change compared to those that change much more. And it turned out, and it has been very useful in all fields of biology, that we can read out a lot of functional information about how the genes function, actually where are the genes, what are their most important parts, and so on and so forth. So the hypothesis or idea, which has started recently to explore, is whether we can use in a similar manner comparisons of metabolomes across different species to learn something about which parts of metabolism that natural selection cares most about. So here we took, actually the metabolome profile was measured for a lot of different mammals in a previous study. It was to identify like longevity-related metabolic changes. And we used this data to identify kind of those metabolites that are the most conserved in the metabolic network. And it turns out that these comparisons make sense because disease metabolites that are known to cause inherited metabolic disorders like phenylketonuria and these kind of things are overrepresented in these conserved sets. So indeed, what matters for evolution also has some disease relevance. So now, like a future goal is to somehow use this evolutionary information in interpreting what is called personal metabolome profiles to predict disease risks. So it is now becoming a big business to measure metabolome profiles, the metabolite concentrations from blood of actually apparently healthy individuals, like all of us could go to these services and get our blood profile for their metabolome maybe every few months or so. And then one can take a look at this profile and ask whether there are some changes compared to the population average. And more or less that's the method that people use these days to figure out whether one has a higher or lower risk of disease. Now our idea is that probably we could make use of evolutionary information to focus only those changes that really does matter for natural selection. So the end goal is to come up with a disease risk score that incorporates these evolutionary information. So my lab looks a bit like a metabolic network here, and this is not a coincidence because it's a network of young, talented people who work together on various topics, and the larger part of the network is devoted to data analysis and computational biology, and a smaller to experimental biology. If you want to learn about not just our lab's research, but also other kind of network science and systems biology research, then please visit this website. You might find some useful information there. And of course, we have a lot of collaborations, both within the Institute and internationally. I would just like to highlight one here, Markus Laser, who is an expert on developing cutting-edge metabolomic techniques to measure metabolites in the Francis Crick Institute, and we have a good connection. So actually some people from my lab spent longer periods in his lab to get trained. So with that, I would like to thank your attention.